In June 2023, a group of researchers from Cambridge University and the California Institute of Technology, led by Professor Magdalena Zernicka-Goetz, announced the creation of synthetic human embryos from stem cells without using eggs or sperm. These model embryos resemble humans at the earliest stages of development and include cells that typically form the placenta, yolk sac, and the embryo itself. Although they lack a beating heart and the beginnings of a brain, they can provide valuable information about the impact of genetic disorders and the biological causes of recurring miscarriages. However, this achievement has sparked serious ethical and legal discussions. In most countries, including the United Kingdom, the law does not regulate work with synthetic embryos, which highlights the need to develop new rules and guidelines in this area. In September 2023, Israeli scientists from the Weizmann Institute of Science reported the creation of a model of a human embryo from stem cells, indistinguishable from a real embryo at 14 days of development. This structure even produced hormones that give a positive result on a pregnancy test. Despite these achievements, it remains unclear whether synthetic embryos can develop into a full-fledged organism. Previous experiments with synthetic mouse and monkey embryos have shown that after implantation in the uterus, they did not develop to a viable state. The creation of synthetic human embryos without the use of eggs and sperm could radically affect the direction of human biological evolution. This achievement challenges the traditional mechanisms of reproduction that have determined the evolutionary development of the species for centuries. If these technologies become widely available, then natural selection, which has shaped the human gene pool for millions of years, may be significantly weakened or even replaced by artificial selection. One possible consequence is that people will be able to completely control the process of creating new organisms, eliminating the inheritance of genetic mutations and diseases. This will open the way to the creation of ideal embryos with predetermined characteristics. This approach can accelerate human evolution, taking it beyond random mutations and natural conditions, and instead moving to the purposeful formation of new properties. However, this could also lead to the creation of genetically modified lines of people, where natural genetic diversity and randomness will be replaced by purposeful decisions. Evolutionary adaptation, which has historically helped humanity cope with environmental changes, may be undermined as artificially created embryos may not have the flexibility to unpredictable environmental or biological challenges. Another significant consequence could be a shift in the boundaries between human biology and technology. If technology allows the creation of embryos with new, previously non-existent properties, this could initiate a new era of post-human evolution, where people become not only a product of biological processes, but also of technological intervention. This creates a risk of inequality between natural and modified people, which could affect the social structure and give rise to new forms of discrimination.